All right, it's been a minute, but we're so back. On today's episode, I'm joined by Dr. Senjuti Saha, Deputy Executive Director of the Child Health Research Foundation in Bangladesh. Dr. Saha is a leading figure in global health and genomic research with a focus on advancing science and public health in low and middle income countries. Let's jump into this conversation about her groundbreaking work and the impact it's making worldwide. I was watching your talk at Gates Foundation, and I was surprised, or maybe not, I don't know, to learn that both of your parents were also microbiologists. So this is like a family business to you almost.
Absolutely. Microbiology is in our genes.
Yeah. And so how does your father inspire you to do the work that you're doing, which we're going to learn a lot about today?
Absolutely. So both my parents, I think, have a huge role to play in what I do today, how I think about problems. My dad is a scientist who is like extremely curious, and since I remember him, he always talked about problems, but problems that were very tangible, problems of people in our community, in the hospital that he worked in, or the clinics that he worked in. And every evening he would come home with these stories, and then he would also share with us how he would solve those problems and what the society needed, what the country needed. And I think my mom really complemented him very well. My mom was more on the prevention side of problems, where she was leading a vaccine lab in the country, but she was also very focused on ethics. So like, okay, this is a problem, we need to solve it, but what is the most ethical way of doing it, ensuring we don't cause harm? So I think it was both of them who has like inspired me a lot.
Okay, so you finished your PhD here at the University of Toronto, and then what happens next? What is the the timeline from you finishing your PhD and then flash forward to you establishing your organization in Bangladesh?
So I finished my PhD, but when I was kind of wrapping up, it takes like 6 months to wrap up your experiments, write your thesis. As all of that was happening, I think I really started questioning, what do I really want to do with my life? I was working in an amazing lab here, productive work, but I just didn't know how my work here would ever reach the community where I grew up, reach people of Bangladesh. And I often thought about my parents who were still in Bangladesh, working for the people, in the country, with the people. I always felt kind of jealous that they're still there and contributing to the society. What am I doing in this, air-conditioned, four-walled rooms? So I kind of had decided that I wanted to go back, even if it is for a short amount of time, spend time with them, understand more the kind of work they're doing. So I started looking for opportunities. I applied for a couple of grants to go to Bangladesh and work there. And then we did get a grant almost, and I had made the decision of going to Bangladesh. But at the same time, in the 11 years I was here, I met my husband. We adopted our first cat. So it was kind of difficult to make that decision of just leaving everything and going. So I think I was trying to balance. I took up a position at Hospital for Sick Children, which was part-time. That would also allow me to work in Bangladesh and kind of do back and forth. And I did that for 4 months, and then I realized it was impossible to do that. The countries were too far apart. The time zones were just not working out. Then I moved to Bangladesh, but then I realized that the grant that I was supposed to get wasn't the best fit for me. So we didn't end up working on that grant, and I started getting more and more involved in Bangladesh. I was still part-time because I did want to come back as often as possible to my partner here. I realized that I had no training while working in Bangladesh on epidemiology. and statistical modeling. So then I decided to do work at Stanford University with a very, very good epidemiologist there, Professor Jason Andrews. So then I spent kind of 1 year at Stanford, but back and forth. Again, most of my work was based in Bangladesh still, but I learned tremendous amount of epidemiology from that time and from Jason. And then, and after that, I was full-time in Bangladesh.
Cool. And you mentioned something about you almost getting a grant, but it wasn't the perfect fit. Can you elaborate on that?
Yeah. So the grant that we had written was basically a grant to use unbiased RNA metagenomics or some kind of unbiased metagenomics to understand what causes meningitis. So the samples would be cerebrospinal fluid from children who had signs and symptoms of meningitis. And the grant was such that there will be a collaboration between our organization in Bangladesh with another organization in a different country, which I would prefer not to name, where samples would be sent.
So when you say your organization, this is the one that you're currently involved with, or something before that?
Yeah, so this is the Child Health Research Foundation, CHRF. This is actually an organization that was established way before I went to Bangladesh. So my father was involved in this organization, continues to be involved. So it was founded by a group of clinicians and microbiologists who are very passionate about children's health. So this is a not-for-profit foundation, research foundation in the country. So that grant was supposed to be between CHRF and this other organization. And the idea was samples would be sent to this other organization. We will do metagenomics. Someone from Bangladesh would also be trained. And everything would be in English, and technology would be transferred back to the country. And while there was like initial verbal agreements, when we were about to sign the agreement, we realized that it's not that straightforward. The other group did not seem very keen on transferring their technology or training or even ensuring everything is in English. So I personally felt very uncomfortable, and this didn't go forward.
And would you say this event impacted your future decisions about collaboration in any way?
Definitely. I feel like this was the first incidents where there were conversations initially, but they were not reflected in papers that we were going to sign. So I'm definitely a lot more careful now. And I also try to do a lot more research on people I collaborate with beforehand. And I'm also more upfront now, I think, than I used to be before. I think every event has some impact in our lives, and it's all about how we learn or take lessons from there and move forward.
Yeah, and I want to move on, but just I don't know. I keep coming up with side questions. So being upfront, what are the pros and cons? Do you think it's more productive, or what has been your experience?
I think, of course, everybody has a different way of dealing with situations. I always say that I am not a mind detective, and I don't expect others to be a mind detective either. So my strategy is always being as direct, honest, and transparent as possible. Now, there are different ways of doing that. I think when I first started my career, I was as honest and transparent as I am today, or I am as honest and transparent as I was then, but I'm more diplomatic now. So the way you express, the way you say those direct things can vary. And when I first started my work in Bangladesh, especially in this field of global public health, I wasn't as diplomatic, and I also did not do my background research. Now, for some people, I will know that, our values are different. So these are probably people I will not even engage in a conversation with. At that time, I didn't know how to do that background research. But I am definitely a person who is very, very upfront and do not want to waste time. Have a conversation, figure out what we want to do together, whether there are alignments or not, move forward.
Cool. Okay, so let's go back. You mentioned metagenomics. What is metagenomics, and how are you guys using it in your organization?
So metagenomics, in very lay terms, is like sequencing the shit out of things. In our case, I am very interested in understanding what causes meningitis. So meningitis is an inflammation of the meninges, which is, a layer that covers our brain and spinal cord. And if there is an infection by bacteria, virus, or fungus, then it does cause a lot of health issues, as you can imagine, because there should be no bugs in our central nervous system. When meningitis happens, the way of testing what bug caused meningitis is by taking a little bit of the cerebrospinal fluid, or CSF, and then you basically test it for the presence of the pathogen, right? Bacteria or virus, you can do culture, PCR, etc. However, in many cases, these traditional techniques of pathogen detection doesn't work. And they become these mysterious cases where we know these are meningitis, but we don't know what caused them. So to solve these mysteries is why we want to use metagenomics. And metagenomics is basically a way of taking the cerebrospinal fluid sample, or CSF sample, and sequencing every bit of nucleic acid in there. Because if there is an infection, the bug is going to be in the CSF, and if you sequence the shit out of it, you will also end up sequencing that bug. However, there are two different kinds of metagenomics. There can be RNA metagenomic sequencing or DNA. Bacteria has DNA. We have DNA. Many viruses have DNA. However, there are also viruses we know that have RNA. And we know that every organism that has DNA at some point in their life cycle will be making RNA. So we decided that we will use RNA metagenomics because it gives a more complete picture. So we use unbiased RNA metagenomics. I've used another term, unbiased. Unbiased because we do not specifically target to enrich any of the pathogens that might be in there. We just take all of the RNA from the CSF samples, whether from humans or from the pathogens causing the disease, and sequence everything without enriching or amplifying for anything. So it's a very unbiased way of looking at what pathogen might be causing that infection. So you take all of the RNA, you make sequencing libraries from them, and then sequence. And after that, you do bioinformatics to look at what are the reads that have come from a pathogen. So you get rid of all the human reads and just look at pathogenic reads.
And how common was this technology in Bangladesh before you guys started implementing it?
To the best of my knowledge, this was never done in Bangladesh before. So this was the first unbiased RNA metagenomics lab that we set up.
And how is it related to your research in your PhD? Is it like, I don't know, is it like kind of like based on your research, or like was it the technology that you were used to work with in your PhD or something?
So I did my PhD in molecular genetics, and I worked also on infectious diseases. I worked on the specific gram-negative bacteria, a dangerous bacteria called Pseudomonas aeruginosa, which is everywhere, but in some cases can cause severe diseases. And I was interested in finding novel alternatives to antibiotics. And in that process, I learned, of course, a lot of molecular techniques. And one of the techniques that I learned was whole genome sequencing of bacteria and doing bioinformatic analysis of whole genome sequencing. So while I did not do any metagenomics per se in Toronto during my PhD, I think all the techniques that I learned in the lab became very useful. That knowledge became very, very useful when we were setting all of this up. I obviously needed additional training, so I spent quite a bit of time at UCSF, at Chan Zuckerberg BioHub, who were my first collaborators to set up metagenomics. But I think PhD just teaches you so much more than just techniques. That has definitely helped me set all of this up in Bangladesh.
And but how did you manage to get this sequencing machine or this technology to Bangladesh?
So when we first started metagenomic sequencing, we weren't even sure if this is a technique that is going to work for CSF samples, especially samples that have been already collected and stored in a freezer. Like, are they even okay? Is the RNA even intact? Can we sequence it? And once we sequence, will the data be interpretable? So actually, in the first phase of the project, the samples were shipped to California, to UCSF and Chan Zuckerberg BioHub, and we did all our sequencing at Chan Zuckerberg BioHub, all the analysis using their platforms, which we continue to use. It's called IDSeq. IDSeq, the name has now changed to CCID. So all the initial phases of work were done in California, and it was super successful. Once the data looked good and clearly all our runs were successful, it also showed that our CSF samples are still good quality. Our donor, the Bill and Melinda Gates Foundation, was interested in setting a small scale sequencing facility in Bangladesh in our lab. So our first machine was a short read Illumina sequencer called iSeq 100. It's a very tiny machine. You can just put it in the back of a truck, take it anywhere, plug it in, and sequence.
How much is it?
It's I think about $20,000 to $25,000. The price varies depending on where you are in the world. If you're buying it directly from Illumina, you get one price. But when we work in countries like Bangladesh, where we don't have the company's office, we have to work with local distributors who work as middlemen, who buy the machine and then give it to you. So they can pretty much charge whatever they want. So actually, it was very hard for us to get a good price because we knew the price wasn't that high. However, to ensure that we also get a good price, and I mean, the local distributor also needs to make some profit, right? So we contacted Illumina, and our friends at Z BioHub helped us a lot, where they talked to Illumina. They gave us discounts. Our local distributor has been absolutely amazing, who made minimum profit, because they were also super excited for us to set this up. And we finally got our machine, and then we started getting our reagents, and we started doing sequencing runs constantly. Now, while that machine was brought for metagenomic sequencing, so much has happened since. We have sequenced dengue virus. That machine became the first machine in the country to sequence SARS-CoV-2. So it's a machine that pretty much our entire lab worships. It's in a very special room. It has its own shrine. But our genomics facility has really grown. We have, bigger machine now. We have another iSeq 100 that we just use for training. So we have come a long way, but that is still our favorite machine.
So when you said you were not sure if metagenomics was the right tool for this problem, you mentioned was it because the quality of samples, or more the method itself? And did you have any alternatives in your mind?
Very, very good question. So I think the first thing that I was most concerned about was the quality, because we know that RNA is not very stable, right? So these samples are collected in hospital wards at room temperature. and then they're brought to the lab and goes through like multiple tests before they can be aliquoted and kept in a -80 freezer. So I was very, very worried about RNA. So quality was definitely one thing. And also at that time, the technology was quite new. And
What year is this?
we started our conversations, I think, late 2017, early 2018. And there was a lot of criticism of the method saying, you can sequence air and you will see something, right? Does it mean it's the cause of infection? So that is definitely skepticism that we also had. Just because, you sequence so deep and pull out, reads of different pathogens, does it really mean it is in the CSF causing infection? So that is something I was also very worried about. So we were able to control for that. So the first time when we selected our samples, we had a lot of positive controls and a lot of negative controls, and see whether those data matched to what they should. And based on that, we moved on to interpreting our mysterious samples.
And did you have any alternative methods in mind, or?
The only alternative to that, in my head at that time, if it failed completely, it would be designing a bunch of different PCRs to look at different pathogens in a biased way. Now, that is actually not a very uncommon way. There are many groups that do that. They have panels of PCRs that they use to look for certain viruses, bacteria, fungi, etc. But the caveat of a method like that is you have to know what you're looking for. But in our cases, we had no idea what we are looking for. So hadn't, metagenomics not worked, it would have been very difficult to figure out. And we tried both, actually, initially. We tried both RNA and DNA, and RNA worked better.
And the last question on this, can you also briefly talk about the data collection and like how did you put together all the samples, and were you faced with any like ethical challenge?
I think this is where institutional support comes in, right? So the way our foundation is set up is already to do surveillance for pneumonia, meningitis, and sepsis. So there was already a study ongoing that was supported by the WHO to collect CSF samples and run some tests. So we had, proper ethical approvals in place. We had proper protocols in place. Written consents were being taken from patients. So that is something I think I'm very lucky I didn't have to worry about at all. It was already all in place. I just could focus on the science.
Great. And so that was a real discovery because you guys had no idea, and like everything came out of this metagenomics study. And was that published in a paper?
So our first results led to some exciting discoveries. In the first set of metagenomic sequencing that we did of CSF samples, we started seeing this virus, arbovirus, called chikungunya virus. It's a virus that's carried by mosquitoes and can causes very high fever with severe pain. And what we saw basically in this large number of cases, the virus could also cross the blood-brain barrier and cause meningitis. This was the first direct evidence of finding the virus in the CSF and being able to look at its whole genome. So that was super exciting. And yes, it finally did get published in MBio. That was my first, genomics paper. So we were super excited.
What do you think are the biggest challenges low and middle income countries face when it comes to public health research?
the first challenge, I think it's across the world, but more so for low and middle income countries, is funding. We just don't have enough funding, and we are really dependent on international donors. And of course, our countries have a lot of competing priorities because we are still developing, infrastructure, building roads, ensuring every kid has access to vaccines and education. So we can imagine it's very hard for our government to support research. So we're dependent on international donors. So funding is definitely a very big challenge. Having said that, personally, I think expertise or lack of expertise is also an equally big challenge, because let's say I get a huge grant of $100 million to do a research project in the country. It's going to be really difficult to find expertise who will be able to conduct very, very good quality research that's a combination of, laboratory work and public health work. Is the kind of work that we usually do at CHRF. And, in public health, it's really part of applied sciences, right? So you need interdisciplinary fields. You need microbiologists, technologists, you need data scientists, you need mathematicians, you need ethicists. And getting all of those experts together in the country can be very difficult. And also, countries like us suffer from severe brain drain, where people are very likely to leave for their, higher education, and oftentimes they do not come back, and it is really the best who leave, which makes work very difficult. Other than money and expertise, things that we really struggle with is something that we already touched upon: access to resources. So getting reagents, machines, supplies is very difficult. It takes a lot of time and paradoxically more expensive in our countries. So makes everything much harder. And, as scientists, we don't really have much money. What we have is time. And when that time is wasted waiting for things, it can be very, very painful, and it can be very demotivating. And I think the last thing that affects us a lot is access to literature, scholarly literature, right? Like when I was at the University of Toronto, I never worried about accessing a journal or an article in a journal. And suddenly I go into Bangladesh, I'm like, oh my God, I'm reading these abstracts, but I don't have access to the paper. And, let's say you sit down, try to do a lit review, and for like half the papers, you can't access it. And you're like emailing a friend or friend of a friend. Can you send this paper, who is in a different time zone, and probably email you the next day. And by then you've like lost your
momentum.
Yeah. So I think that's also a very big barrier.
Yeah. You said something very interesting, that in science we don't have money, we have time. I like that. So I did my undergrad in mathematical biology, and it was around the COVID time. So we were doing research on inequalities in vaccine distribution around the world. And one of the few things that I remember to this day was this thing that they mentioned, like the rich countries, they just they prioritize vaccinating like their own people, which to a degree makes sense. But they were like, the argument was like, you keep ignoring people in other parts of the world, and the virus evolves there and just brings out more variant that ultimately makes your vaccination ineffective. So I'm just wondering, and that would be a motivation for people in West to donate vaccines to other people. I'm just wondering, what would be the motivation of international donors? Is it just empathy, or they're interested in the results as well? I'm not sure if I'm making sense.
You are definitely making sense. The inequity that we have seen in vaccine distribution during COVID is appalling. But at the same time, probably not so surprising. This is not the first time this has happened. It has happened again and again for other vaccines. An example is a Haemophilus influenzae type B vaccine or HIP vaccine. That, for example, came to Bangladesh 21 years after it was introduced in the US.
Thanks to your father, I guess.
Yeah, he did definitely help in facilitating introduction of the vaccine based on evidence that we generated in the laboratory. It's a responsibility of our global leaders to ensure that everybody has equitable access. During COVID, it was not only they were vaccinating their own people, there were countries, including Canada, who were hoarding vaccines, right? And these vaccines eventually expired and had to be thrown away. So this is responsibility of, I think, countries with resources to ensure everybody has equitable access. We have to protect the entire world. And if it's a selfish reason, like, to protect myself, I have to protect my neighbor, then so be it. But I think there is a lot more than that. Not everything can be selfish, right?
Yeah.
We all have responsibilities to ensure everybody has access to basic prevention methods.
Yeah, and I think after COVID, there's been a lot more attention on public health research. What role do you think cultural context plays in scientific research, especially in the field of public health?
I think cultural context is extremely important, and we must acknowledge that it differs. Countries to countries, regions to regions, and even within a country, different places can have different cultural context. For example, public perception of science, public perception of scientists, right? So, as an example, Bangladesh is a very pro-vaccine country. We have very, very high vaccine coverage, reaching almost 95% across the country, even in hard-to-reach areas. And this is because the public and the mothers, especially of the communities, are very knowledgeable that vaccines are needed to ensure their children are healthy, and vaccines are good for the society, for the child, and for the neighbors, right? Not the same case in many other countries, even in developed countries like the US, where we know now that there are communities who are very anti-vaccine, right? So we will work how, where, what will be our priorities really depends on what communities you're working with. Now, there are communities who are very worried about taking vaccines that, let's say, come from North America. And, these could be some countries in Africa who have seen that every time, white people came, black people were put on a ship and taken away. They have, historical reasons not to trust.
Yeah.
Right? So I think, how we work with those communities are going to be different. So cultural context really matters. And one of the reasons I moved back to Bangladesh is also because of the cultural context, right? I grew up there. I kind of understand the culture. I speak the language. I understand, perception towards science. So that's, I think, a privilege I had, and I wanted to leverage that privilege. I could be doing the exact same thing in a completely different country, and chances of me being successful is not very high because I do not understand the cultural context.
One of the most exciting things that you're doing besides science is this initiative called Building Scientists for Bangladesh. Can you elaborate on that? What is your long-term goal with this program?
Building Scientists for Bangladesh is an initiative that we started in 2022. It's a completely bottom-up initiative and super exciting. I think it's the most rewarding work that I do with my team. So it was during COVID, and I mentioned earlier that our lab was the first to sequence SARS-CoV-2. And what that did is we suddenly got a lot of media attention. So we already had some popularity, but this really, during COVID, when one virus is everything that everyone talked about, we became kind of this popular faces on television, people from our team. And I showed up on like a lot on TV. So as people like started watching, I think this woman who is doing science in Bangladesh, it kind of piqued interest. And in a very positive way. So I started getting a lot of messages from mothers saying, can my daughter also become a scientist? What do we need to do to ensure she can be? I started getting a lot of even handwritten letters from young girls saying, we've never seen a scientist before. How can I become a scientist and actually work for the country, for health? And I realized that is so true, because when we go to a hospital, we see a doctor, we see a nurse. We know buildings are built by engineers. We see them. We see a plane. We know it's being flown by a pilot. We see cricket. We know someone's playing cricket or football. We don't see scientists because scientists do work behind curtains. They are in buildings usually. And I think also in our culture, whenever we think of scientists, because of our education, we kind of think of, white men with long gray hair and glasses are the only ones who can become scientists. So, when I realized the privilege I had, that I have two microbiologist parents, and because they were always super busy, and I always, from school would end up in one of their labs, either in my father's microbiology lab or my mom's vaccine lab. So I had very early access, which 99.999% of the people don't. And the reason I'm here today being able to do so much work, because my parents opened up their labs to me. And after getting those messages, I realized I have a responsibility. Our generation's job, who are working at CHRF now, it's our responsibility to open up our labs for them and ensure they get the same access and exposure. Meet scientists, talk to scientists. So we started this initiative. We launched it in 2022, Building Scientists for Bangladesh, or BSB, and it has 3 streams. The first 2 streams is for school students.
Just to clarify, the program is just in person.
The program is right now all in person, yes. So the first 2 stream is for school students. So in the first stream, which we call Bringing Science to People, we travel across the country and we set up science camps. We take a microscope with us. We take, petri dishes with us, even petri dishes where you can see zones of clearing for different antibiotics, so you can learn more about antimicrobial resistance. Kids bring their own yogurt. They bring banana, and we extract DNA together. They look at bacteria in their yogurt. They do, fingerprints on petri dishes before washing hands, after washing hands. The next day they come back and count the number of colonies. So really it's like experiential learning, right? So we have done that across different schools and reached over 10,000 students.
Wow.
And the next stream is called Bringing People to Science. So it's also for school students, and they have this option of coming to a research lab and spending time. So they could pick their favorite lab, whether it's biochemistry, microbiology, molecular biology, sequencing, whatever they want, and shadow. Right? But also do like simple experiments, similar to what I explained for the first stream. And really, the way we design all of this is kind of learning through experience and hands-on activities. There are no exams, no tests, but designing games. So, in Stream 2, for example, we have a game called Infectious Disease Detective, where they learn a little bit about how work is done in the lab, and every day they receive a clue. And they have to solve a mystery. And it's like a patient has come into the hospital and the patient has fever and a blood sample was taken and these were the tests that were done. Now figure out what is the culprit. And by the end of their program, they not only have to figure out what's the culprit, but also come up with the right antibiotic to bring justice to the child. So it's like a video game format, but in person and with clue cards. So it really makes everything like super exciting. And then the third stream, we call it developing skills, continuing education. And this is for people who are already in university or have graduated or, way beyond graduation, maybe even retired, but they want to get, access to current technologies. And they can come spend time in our sequencing lab, in our next gen, or like bioinformatics lab, where they just come, they spend time, pick their bacteria or virus, sequence it, look at the data that comes out of it, and learns how to analyze that data. So that's a building scientist for Bangladesh. But again, it's not meant for theory, right? You're enhancing theoretical knowledge. I think schools do that, universities do that, and more importantly, there's a lot of resources out there online to get those theoretical knowledge exposure. I think through BSB, what we do is we give this experiential exposure.
Yeah, I like that you go around the country and do these things for kids. And I think that's also a thing you do with your research, where you don't rely on people coming to hospitals anymore, and you send people around the country to do surveys and stuff like that.
Absolutely. So there are two things that we do, or two examples that I can think of right now. So in low resource countries where people are still paying out of pocket or there isn't a lot of access to healthcare, a large proportion of patients, especially children, do not end up at the hospital. They will go to a community clinic, perhaps. The parents will go to a community clinic and just get medications and treat the child at home, or to a village doctor. When we think of research studies, like traditional research studies, we only think of research studies conducted in hospitals. But you can imagine in a country like Bangladesh, where majority of sick people are not even coming to the hospital and you're only doing studies in the hospital, you're massively underestimating burden of disease. So it's really important that we also do work in the community. So there is this one rural community that we have been working with for more than 13 years. Actually, we have been working with this community since 2007, so it's been a long time, where we have a group of more than 100 community health workers who are mothers from that community, visit every household with an under 5 child every week, and look for signs of pneumonia, meningitis, sepsis, or other illnesses. And if the child is sick, we record the illness. If the child has taken medications, we record that. And if we think the child needs care, then the child will be referred to the nearest hospital where our doctors are there to see the child. So, we can really link community data to also hospital data if the child goes to the hospital, and if any lab tests are done, we'll also have access to what lab tests were done and what results we can find. So when you have this complete picture, you can kind of extrapolate and model what is the true burden of diseases and severity of diseases, what is true mortality, etc. That's one thing that we have been doing for many years. The second example that I can think of is this countrywide surveillance study that we are doing. And this is very much linked to the work that I have done at University of Toronto, molecular genetics. So during my PhD, I told you I was looking at alternatives to antibiotics, and those alternatives that I was looking at was basically tails of bacteriophages. So some bacteria produce particles that look like tails of bacteriophages that kill other bacteria. So in that process, I learned a lot about bacteriophages, which are viruses that kill bacteria. So we are actually doing this study across the country where we are collecting water samples and looking for bacteriophages that are specific for this bacteria called Salmonella typhi that causes typhoid. Now, typhoid causes bloodstream infection, and we want to understand across the country how many kids have typhoid, what is the burden of typhoid across the country. But we cannot take blood from every child. We cannot do community study everywhere. So what we're doing is collecting sewage sample because it's a waterborne disease. And in those sewage samples, we're basically looking for viruses that kill the bacteria or phages that are specific to Salmonella typhi. Because if we know that there is phages, there will definitely be the bacteria. This is a bacteria that's very hard to grow in the lab. So we are looking for phages. And we are basically making this heat map of the entire country and finding hotspots of Salmonella typhi circulation. So this has been ongoing for 2 years, and we have an amazing group of people constantly traveling across the country, collecting wastewater or sewage sample and shipping it to the lab, and we're testing it for the phages. So that's the other type of work we do, which is super exciting.
Yeah, and to me it sounds like you have a whole established network of doctors and nurses and scientists. So is it like in parallel to the healthcare system, or you guys are part of the Bangladesh healthcare system, and how does it work?
So we are a not-for-profit private organization. Of course, any data that we generate are open, accessible. We will report to any relevant ministry or health institute, government health institute. And there are certain diseases that are reportable that we will report right away. For a lot of the work that we do, if it's supported by WHO, then we will immediately share all our data with the WHO. So as I said, Bangladesh doesn't have a lot of resources to support work like this. So this work is usually supported by international donors, but the data is all available for local policymakers.
And speaking of WHO, you're involved with several WHO advisory committees and boards. So how does this work influence what you do at the national level in Bangladesh?
I would say it's kind of the other way around, where I think I'm in a lot of boards to bring that local context to the table and a lot of these advisory committees. For example, I am in, Salmonella Vaccine Advisory Committee. So local context of what's happening in the country right now. Also, because I work in the field of Salmonella, what do I know from working with other groups about burden, about antimicrobial resistance, sharing my knowledge and expertise, at the same time learning about what's happening globally, and perhaps saying what might work in Bangladesh if we implemented a certain policy or introduced a certain vaccine, right? Now, oftentimes what happens in those global meetings do not necessarily dictate how I work in the country. Rather is the other way round.
And well, I would say with the attention on public health from COVID-19 pandemic and everything after that, what's something that you think the world still hasn't learned?
I think we still haven't learned that we need to learn. I think that is my biggest takeaway. We, as humans, we forget and we refuse to learn from history. There will be another pandemic. And I see a lot of people and experts saying, pandemics come every 100 years. That is not going to be true. You know, rate of pandemics, rate of epidemics are increasing as global warming becomes worse, as we encroach into natural habitats of animals more and more. There will be more chances of new viruses, or even, other pathogens perhaps, to come into the human population. We travel a lot more now than ever before. So chances of these big outbreaks happening are getting higher and higher. And I just worry we are just not doing enough to ensure that we catch circulations early enough, that we prevent pandemics or next epidemics from happening. I don't think there is still a lot of work that people of different fields, right? So people who work on climate change, people who work on microbiology, infectious diseases, or pandemics. I don't think we are talking to each other enough, because all of these things come together to cause a pandemic. That's a very, very big fear I have. Also, I don't think we are focusing enough on surveillance either. There are all of these initiatives to prepare for pandemics, but there is no way we can prepare for pandemics if we do not know what is circulating right now. So we talked about meningitis in Bangladesh, for example, right at the beginning. And I was telling you how there are all of these infectious cases where we do not know what pathogen is causing it. So when you do not know what is baseline, how do you figure out an uptick? How do you know what's unknown when you don't know what's known? So I think there needs to be a lot of investment in figuring out what are the pathogens that are circulating now, and doing a lot of mathematical modeling to figure out which pathogens that are circulating now have the potential to take off, and constantly looking at also human-animal interface.
Yeah, and I think you kind of answered my next question too. I was gonna ask about how can the global scientific community better support your efforts in Bangladesh, and if you want to briefly comment on that.
I think there are many ways of working together, and there are many ways of supporting a lot of the amazing work that's happening across the world. The fact that I'm here at University of Toronto talking to you, doing a podcast, is already global support. So, enhancing visibility of the work that's happening. Global support could be exchange programs, right? While we run a training program in Bangladesh, we could also set up training programs across oceans, right? That could be global support. Global support could come also in the form of money, funding, ensuring that these programs are more supported, whether it's surveillance or training, and more knowledge is generated. Global support could be getting groups together from different parts of the world and kind of catalyzing changes or thoughts such that more programs can be set up across different countries by local scientists. So I think there is a lot that we can go do together.
Okay, you said about the possibility of future outbreaks. So in your work, how do you prioritize what project to work on, what type of disease to prioritize in your research?
So in our group, we focus on diseases that are most common in children. So, neonatal sepsis, so bloodstream infection in babies who are in their first month of life. We focus on pneumonia, still the largest killer, the biggest killer of children across almost all resource-limited settings. And then we focus on meningitis because it's severe disease, burden is very high. can lead to both death and disability. So we kind of focus on the diseases, and then under that focus on the most common pathogens that cause these diseases. So let's say for neonatal sepsis, we do a lot of work on Klebsiella pneumoniae, a gram-negative bacteria that causes antimicrobial resistant infections. For pneumonia, we focus on Streptococcus pneumoniae, we focus on RSV. For meningitis, Haemophilus, Streptococcus pneumoniae, Neisseria, and a panel of viruses. So really, a lot of our work is guided by what are the most common diseases. Having said that, we also see a lot of epidemics in our countries, right? Like dengue outbreak, chikungunya outbreak. Hospital beds suddenly become full of patients with these diseases. So oftentimes we have to shift gears and do what is needed for the country, for the global community at that time to generate evidence to bring about policy changes.
Yeah. So as someone who's worked in both high and low resource settings, what are the biggest misconceptions people have about conducting research in low-income countries?
Very short, I think the first misconception is people are less smart in poor countries. If you don't have money, you don't have brains. So that's, I think, a huge misconception. And also another misconception is big institute means big intelligence. You could be in a very small organization, but have very smart people working.
You mentioned earlier that Bangladesh is a very pro-vaccine country, which I'm not going to lie, kind of surprising when I compare to my own country in Iran. I mean, the majority of people got vaccinated, but there were, I guess, a vocal minority about like anti-vaccination, and they shared the same concerns that this is coming from the West. We don't know what they have in mind. So what's your perspective on vaccine development and distribution in countries like Bangladesh, and what challenges exist that may not be widely understood?
I think one of the biggest reasons of disparity and the inequity in vaccine distribution that we were talking about before is this centralized idea that all vaccines will be produced in a couple of these places by a couple of companies, and then it will be distributed across the world. That cannot work. If the system stays centralized, we will keep on facing the same obstacles again and again. It's very important that these systems or vaccine development is decentralized, distribution is decentralized. And I think a lesson that we really need to take from COVID is, technologies can be decentralized. Now, with, for example, the mRNA technology, I know there are initiatives to ensure that more countries can quickly produce these vaccines and distribute them. I think a lot of focus needs to be put and resources need to be put into developing infrastructure and expertise in every region. Every country will not be possible in every region to ensure there's local capacity to produce vaccines, to get the vaccines quickly to who need it the most. This not only ensures, vaccines are distributed during global pandemics, this also ensures that there is capacity strengthened to ensure that when there are local outbreaks, there can be vaccines locally manufactured and given to people very, very quickly. And when you have access to technology, when you have technology at your fingertips, there is more chance of innovation because people working in those facilities in resource country settings know the best what is needed. So they will understand the problems and really use that technology to come up with solutions to the problems. So I think decentralized vaccine manufacturing is the biggest thing we need right now.
And I have like one big picture question, I guess. So you were a PhD student, and you transitioned into this like amazing career, and you this leadership role that you have. So how was this transition from like basic science, I guess, to more practical and leading an organization? And then the other way around, now that you are uh, doing all these amazing stuff, how do you find time to like keep up with the latest science, and like what is your process uh, for getting research from lab into peer-reviewed journals? You've published over 75 papers. So how is this like link? Like how does it work for you?
So I'll first try to answer the leadership question. When I moved to Bangladesh, I didn't see myself in a leadership role. I was just excited about the science and working together. But I think I've been very privileged and things have worked out. In some cases, I was just at the right place at the right time, where I have been able to, get the resources to build a big team, and we're doing well. And which, of course, building a bigger team means I had to go on a more and more leadership position, and in some cases also more administrative position. I will say it was not very easy. I had to learn a lot about leadership. During my time at grad school, I don't know about now, but we didn't get a lot of training on how to lead teams. So I had to take a lot of courses, and I continue to. So, for example, right now I'm taking courses on negotiations and collaborations with other institutes. Read a lot about leadership, on how to like lead organizations, especially in resource constrained settings, how to keep people motivated. But something that has been instrumental in, I think, what I do right now, or the reason I'm able to do this, is also the amazing mentors that I have had. And I don't have one mentor. I always have many mentors, because there are so many people that have come across in my life who are successful in their own ways. And, there isn't like one person or two people about whom I love everything. There are certain personality traits or characters or strengths that in different people I like, and I make it a point to reach out to the person and be like, I saw this, I am leading this organization in the country, I would love to learn more about your experiences and keep in touch. So and I, to some people I'll be like, will you be my mentor, can I like take regular calls with you? And I continue to do that. And this is so important, and I don't think this ever ends. So, of course, learning from courses, textbooks, but also from other people's experiences. And sometimes you just need people who understand your context and your background. There could be super successful scientists in, North America, but might not understand my context at all. So perhaps not so super successful scientist in South Asia, let's say India, could be a lot more helpful in helping guide me on my next steps, or how to lead my team, how to work with people, etc. So mentorship, very important throughout your life. And then you asked about publishing papers. Oh, publishing's getting harder and harder. Like you see all of these grays. Every gray is a rejection. As you can see, grays are increasing super fast. It's really hard to find time to write papers, but I am still learning, and I think every paper that I am involved in is getting better. I'm becoming a better and better writer, and hopefully I will throughout my life. But what's very difficult in Bangladesh is, because English is also not our first language, is actually having people who can write Papers, like scientific papers, is a specialty writing them.
Yeah.
So that's definitely a very big challenge, and we have to spend a lot of time in getting data together and publishing them, going through that entire brutal process of peer review and getting it out there so people can read our work. It's still very time consuming, very difficult, gives me a lot of anxiety. And also, you mentioned the word 75. You'll notice that a lot of them are basically teamwork, right? So I think as we are doing more and more work in public health and broadening our pathogen list or disease list, we work with more and more collaborators across the world, which also means that we publish together a lot. How do I keep up with literature? It's getting harder too, right? Different people have different ways. I listen to a lot of podcasts. I'm on one right now. So for, the example is like Nature podcast. It's like pretty brilliant. I think I started listening to a Nature podcast through Corona Pod. And then I found others and others. Of course, there is Feedly, but I also talk a lot. So every time I get an opportunity to speak to a fellow scientist, I will try to be like, oh, what's latest thing that you are thinking about, something exciting that you have read about. When I go to conferences, even if I am giving a 15-minute talk, but it's a 3-day conference, I kind of put my laptop away. I do not take work to conferences because I'm like, this is my time to take in as much as possible, because that is where you learn about the latest things that's happening. Because if you're at a conference and just, working and responding to emails, it's a missed opportunity. So those are some of the ways.
And since you brought it up, how do you find good mentors?
How do I find good mentors? So a lot of them come from attending conferences. When I hear someone talk, oftentimes I'll ask questions or maybe meet them at a networking dinner and try to understand their personality, do, hi, hello, and see if this person is even approachable. And then if this person is approachable, then I will send an email and see if they would be willing to get on a call with me. I have found mentors through collaborators, through, small group meetings I've had with, perhaps collaborators or collaborators' bosses, collaborators' mentors. I have found mentors through friends. I have found mentors that I just heard about from others, read about their work, and, reached out saying, I'm this person. I absolutely love your work. I would like to learn more about how you do this work in this setting. How did you build your team? And of course, not everybody is as responsive. I'm getting to a stage when people end up responding to my emails, but not everybody is as responsive. And just because you are a successful scientist doesn't always mean you'll be a good mentor. I'm looking for them all the time.
Yeah. Well, one last question, and then I'll ask you about advice for young people. But before that, so about your organization, looking ahead, what are some of the most exciting opportunities that you see in global health research and in terms of long-term projects for your organization?
I think that our biggest focus right now for the next 10 years or more is to build an atlas of pathogens, right? Building an atlas of all the pathogens that cause the most common diseases. Atlas of pathogens that cause meningitis. Atlas of pathogens that cause respiratory infections. Atlas of pathogens that cause sepsis. I think that's the number one goal. Gradually chipping away at what causes diseases. Basically understanding what are the knowns to figure out the unknowns.
And thank you so much for this conversation and showing up. This is my first episode in Toronto, and I'm just so honored to have you as my first guest here. You've been here for 11 years, so I know Toronto means a lot to you too. So last question, what do you say to young people? What are some life lessons, some advice, and what do you have to say to them?
Firstly, I'm still young.
Yeah.
Secondly, it's very, like, I can't give advices because I think everyone's context is so different. The only thing I think is very helpful to do is do what you love. Sometimes we just get so influenced by what is considered success by our peers or people around us that we forget what will make us happy. What do we love doing? Because if we are doing something that we don't enjoy, that we are not good at, then chances of us becoming successful and putting in the work to be successful is very difficult. And unlikely that we'll put in that much work. So I think it's very, very important to constantly ask ourselves, what do we love doing? What do I love doing? And am I good at that? What is that intersection of something that I love doing and I'm good at, and pursuing that and ensuring that, I become the best in that? Because when you love something, when you're good at something, that's the only way of excelling in something. And it should not be guided by what people around tell you.
Thank you so much for tuning in to today's episode. If you enjoyed the conversation, please take a moment to rate and review us on your listening platform. Your support helps us reach more listeners and keep the discussions going. Until next time, take care.